It's not in here. Bear with me. I came down with a rather massive head cold over the weekend, and hopefully you can understand me and you're sitting clear enough so that microbes don't get transferred as readily as they possibly could be. So I'm going to share with you some work that we've been doing in the lab the last few years. That initially when I thought about this talk, I was going to incorporate a lot more than I really could because of the amount of information we've gained, and primarily from a USDA-supported grant to look at nitrogen aspects of microbiome in collaboration with Matt Liebman, Mike Castellato, and Kirsten Hofmackel. And so I'm going to talk a little bit about this, and by way of stating the obvious, Iowa produces lots of corn. Okay. And maize is a crop obviously of major economic importance. About 40% of the corn is produced in the U.S., and high yields are typically achieved obviously through inputs of inorganic fertilizers. And in most cases, it's due to conventional cropping systems, at least in the upper Midwest, of a corn-soybean rotation. Okay. And so everyone's pretty much aware of that. And Matt has developed these, there's some alternative systems. And the questions are they better or than conventional systems? And this is following on Matt's published work on basically their observation at the Marsden long-term experiment site that you can have in a 4-year rotation comprised of maize, soy, oats, alfalfa, and alfalfa, with composted manure inputs. You exhibit about comparable yields and a reduction in nitrate loss from those systems, okay, from the water. And this is basic things. The manure is applied for maize, so it's done every few years. It's managed more or less to be about the amount of nitrogen, although that's not exactly accurate all the time. But more importantly is the decrease in nitrate leaching underneath those systems. And so, obviously, soil microorganisms play vital roles in, well, I'm biased. I'm a microbiologist with a very microcentric view in everything, okay? And so, when I think of soil microbes in soil, they colonize not just the soil habitat, soil particles, and minerals, they're colonizing the roots, and they're inside plants, both on the roots, they're inside the aerial surfaces, they're on the surfaces of plants. And they can result in both beneficial symbiosis or nutrient cycling interactions, or possibly detrimental interactions where there's pathogens or competition for nutrients that other microbes can't get or the plant can't get. Okay. And obviously soil microbes are key to the nitrogen cycle. And when you think about soil, you have to think about it has lots of organic material, and there's decomposition of this organic material, primarily proteins, by soil microbes that liberates the production of ammonia that can be utilized by bacteria, other microbes, as well as plants. So there's competition for that turnover of nutrients in the soil. And the other thing about microbes that are relevant to this talk really is nitrification. And that is the process by which ammonia is oxidized to, originally we thought was a 2-step process by bacteria. There was ammonia oxidizing bacteria that would produce nitrite, and then nitrite oxidizers would oxidize it to produce nitrate. That is the labile form that's readily lost through the soil profile. What we've discovered over the years is that not only bacteria, but archaea are capable of this transformation. And more recently, the discovery of a bacterium, or group of bacteria, that carry out both steps simultaneously, or in the same organism, rather than being 2 different groups of organisms. And so, these are the ammonia oxidizers, the thalmarchaea. We'll talk a little bit about those in nitrous modales. And nitrate oxidizers we're really not going to talk about today, mainly because of lack of time. So, what do we know about the soil biology of this long-term cropping system, which has been a focus of study by a variety of groups, and primarily Kirsten Hofmackel's group and others, But I'm just going to summarize some of this here. One thing that Keyston's group determined is that protease activity is greater in diversified systems. So there's more extracellular proteases produced, more turnover of that organic material. The soil microbial biomass and microbial metabolic activities are greater by different depths in the system. And I'll just mention that these two assays are very difficult to do, and they're very hard to quantify and to get really good numbers on them, okay? They're just tough, okay? But these are really good estimates in terms of what's going on in the soil. The other thing is through Will Overholt and the Liebman and Castellano labs, is they determine ammonia production rates change are different between the two systems. The amount of potentially mineralizable nitrogen was lower in a depth-dependent manner. And also, there's smaller nitrate pools in these systems. So basically what we've got is a suite of pieces of information that suggests that there's some different microbial processes occurring, or microbial activities. And what we wanted to do was to look a little more deeper into who are these organisms and what's the nature of these organisms and their interactions with between the two cropping systems, as well as with the plants. So our guiding hypothesis was really nothing more than increased soil carbon inputs of the diversified systems foster different microbial assemblages, resulting in a tighter coupling of available nitrogen supply and demand and smaller inorganic end pools. Those are very lofty goals. Did we actually demonstrate that? No. We basically really demonstrated the first one. There's different microbial assemblages, and we've just have been able to tweak apart a little bit about those different communities, those different systems, and more importantly, how they interact and influence plant colonization and development and assembly of a plant microbiome. And that's led to our next question is, does the soil and plant microbiome change over the course of maize development through the growing season? And how strongly do different microbial assemblages influence assembly of the root microbiome? So if you have one microbial community, is the plant such a strong selective factor? It really doesn't make a difference what soil you're in or what habitat you're in, because the plant has such a strong dominant role. Okay. And then the other question that we had, and which we're particularly interested in, is that can we identify who are most likely the most metabolically active in the community? And does the cropping system and or the presence of the root influence these metabolically active component of the community? So this is a rather simplistic view of our experimental system. We're using next generation sequencing based approaches to look at the microbial community, the microbiome, in these various habitats. And we're focusing first on the bulk soil. Those are microbes in what we refer to as root-free soil, usually obtained either between the rows, right in the middle between two rows, or in the laboratory in rhizotrons without a plant. So we could say definitely not a root there. Then we also have different aspects of that continuum between that root growing through the soil. It comes in contact with soil, and these root exudates influence that soil. That's referred to as a rhizosphere. The rhizosphere soil is the soil that is in contact with the roots. Okay. That is distinct from the rhizoplane. That is the bacteria that intimately are on the surface of the root. Okay. The problem and the challenge is it's really hard to separate that 1 to 2 millimeters at most distance between the root and the bulk soil, finding that rhizosphere soil. So we took various approaches. Sometimes we would shake off the bulk soil, take the rhizosphere soil, collect it, and that's what we looked at. And then other times we take the root that's adhering to the soil that's adhering to the root, wash it off of the root itself, so you have rhizoplane and rhizosphere, because it's really hard to separate those. And then the other thing we looked at is our endorhizosphere community that is distinct from what's on the rhizosphere, okay, which is distinct maybe from rhizosphere soil, rhizosphere soil all the way up to bulk soil. And what we did is we isolated either DNA or RNA And we did community DNA or RNA profiling. And you might ask, well, why did we do both DNA and RNA? So we're using amplicon-based sequencing. That is, we're using a piece of DNA as a target for PCR and sequencing using Illumina technologies. Now, that's usually based on DNA. Okay, it's easy. DNA chemistry is easy compared to RNA chemistry. And so what we refer, just for keep terminology straight, we refer to as the total bacterial community, the resident community. That can include dormant cells, quiescent cells, relic DNA. DNA is part of a soil biofilm, and they spew out DNA when they make biofilms in soils. So that could be part of that. Okay. We distinguish that from our metabolically active community, which is comprised of actually extracting RNA. And we focus on, and the nice thing about ribosomal RNA is fairly stable. Messenger RNA is not, okay, but ribosomal RNA is very stable. And so we can use that as a proxy under the assumption that the number of ribosomes reflects the metabolic status of the cell. So a cell that's more or less quiescent might have one ribosome and one 16S ribosomal RNA gene. And if they have 10 ribosomal RNAs to DNA, they're likely more metabolically active. It's a failed assumption from the very beginning. Okay. However, it's the best we've got. Okay, so we can look at the community from two different perspectives. And so we're saying that the abundance of ribosome reflecting the RNA says it's more metabolically active. And we're going to be looking at primarily bacteria. I must say that I am biased towards bacteria. I understand them better than fungi. Okay, maybe it's because of familiarity. But I'll talk a little bit about fungi, but most of the talk will be about bacterial or prokaryotic diversity. We focused on one particular variable region, the V4 region of the ribosomal RNA molecule. We amplified it and we did sequencing. We then did ITS region for the fungal communities. Okay. And for those who are not familiar with how you deal with this information, I wanted to introduce you to the concept of operational taxonomic unit. A concept well developed in this building a long, long time ago, back in when Burgess Manual of Determinate Bacteriology was actually housed here in Iowa State University in this building where they did a lot of the biochemistry of bacterial taxonomy. And they would do what you call operational taxonomy. You don't know what the actual species is, but you use it as a designation of who it would be to differentiate from other ones. So in our sequencing reactions, we would get 15 to 38,000 reads of any particular sample. So that'd be like coming into Ames and doing a census of 38,000 counts, and then using that to get a sense of what Ames is like, and then do that to Des Moines and say, well, we undersampled Des Moines at 38,000, okay? But you get a sense, we can get a sense of the community difference. We do filtering, and when we cluster based on 97% sequence similarity identity, okay? So that's good enough for what we call a species level, and you get a barcode on each one, and you got a unique sequence. And what we do is we match them up and say, okay, these two barcodes have the same sequence. That's operational taxonomic unit 1. Okay, we have around 250 base pairs on the bacterial, and that size, and sometimes a little smaller for the ITS region. And we can then do blast searches essentially against archival databases to find out who or with potential taxonomy of that particular operational taxonomic unit. Okay? So we get all that information, we go through a series of bioinformatic pipelines. I'm not going to talk about all of them, just saying that we do a lot of different ones. Most importantly though, we use linear discriminant analysis, effect size, and DSEC, differential abundance. Particular DSEC is used a lot for transcriptome studies, it's actually derived from that. And then another one is for network analysis. Okay. And what we get is something that looks like this. We get large amounts of data, and for any one OTU, okay, OTU1, we know it belongs to the Protobacterium phylum. It's an alpha protobacterium. It belongs to the order Rhizobiales. It then is in the family of Bradyrhizobiaceae, and it could be the genus Bradyrhizobium. Okay, we can get all that piece of information on that one read, many of which, though, will have a question mark, maybe here, Rhizobiales. Okay, so you can sometimes have classification at various levels of resolution. You lose that resolution as you go further into the taxonomic classification. Okay. Oops. How did I do that so quickly? So that is, we do that for all the different OTUs up to, in this case, OTU 10,249, which was the Veric microbiome. Okay. Which I'm particularly fascinated with, because one thing they've shown in agriculture seems to be the reduction of Veric microbia from prairie systems. Dramatic reduction in that particular taxon. So I'm going to talk about the work done by a former master's student, Cassie Weinberg, who most of you, if not all, know. And she had this guiding hypothesis that she wanted to ask. That is, do the root-associated microbial communities of maize differ the most between cropping systems at developmental stages of high nitrogen demand, or not? Okay, basically. And she predicted that they would be most different between the two cropping systems at vegetative stage 11 and during the kernel development stage. And so what she did, she sampled 4 different times during the growing season and collected the plants, and she examined bulk soil, rhizosphere soil. So she removed out in the field the rhizosphere soil attached to the roots into a bin, and then sampled that and put it on dry ice, and then we took it back to the lab to process. And then back in the lab, we extracted the bacteria that were on the surface to look at the rhizoplane community. And I'm going to give you a lot of information here, okay? And so I'm going to walk you through this. But this is basically an ordination plot of what we call the Bray-Curtis dissimilarities. And so it's taking all those reads from a particular sample and it's asking the question, are all the samples from the bulk soil from conventional similar to all the samples from the diversified bulk soil or not? Okay? And this likewise for rhizosphere soil and rhizoplane. And it's pretty obvious when you look at this diagram that you've got some things here. This in red is the bulk soil. The green is, oops, too heavy of a hand. The green is the rhizosphere soil, and the blue way out here is the rhizoplane. Okay? And this type of separation is basically true both for the fungal and the prokaryotic, the bacterial and archaea community. And you see the separation to varying degrees at all 4 developmental stages. Okay. We also did a statistical analysis using a Primanova, okay, and using Adonis comparisons and doing contrast at 999 permutations to determine whether or not there's a statistically significant difference between the bulk soil in the cropping systems at each time point. Okay? And what we saw was, yes, there is a statistically significant difference in all of these different comparisons, the bulk soil in the 2 cropping systems, the rhizosphere soil in the 2 cropping systems, and the rhizoplane in all these systems with a few exceptions. The only times that the rhizoplane was not different between the 2 cropping systems were at kernel development and physiological 5-7. Okay, this is after a Bonferroni correction, so it's a very conservative test to. So it's possible that's significant, but by the time you get here, it's like a 0.16, and it's clearly not different. And you see that in physiological maturity of fungal. And that might suggest that as the plant develops and ages, the cumulative effect of the plant on the microbial community is stronger. Initially, it's much more the environment the seed bank in the different soil communities that influences this assembly. But as the plant matures and ages, that becomes a much more dominant factor. And we see it maybe a little sooner with bacteria than fungi. And so this is a lot of information. This is actually a lot of, well, is there differences? Addressing part of Cassie's questions, and it's partially answered yes. But there's another observation I want to make. So these were only comparisons between like bulk soil, okay? What about, is the bulk soil the same as the rhizosphere soil? Okay? Now, many of you probably have heard of the concept of a rhizosphere effect, okay? That is the consequence of the exudation of plant nutrients and the physical, as a nutrient source, as well as it alters the physical chemical properties of the root environment, that changes what's there. And the rhizosphere is much more metabolically active. The rhizosphere soil is much more metabolically active. And we were kind of surprised by this observation, particularly for the bacterial. And that is, we saw no difference between the bulk soil and the rhizosphere soil in either the conventional diversified system at vegetative stage 4. We did see it in the conventional system at stage 11 and at kernel development only, but we never saw it in the diversified system. So we asked ourselves, is this a possible sampling artifact? Okay, soil moisture difference, aggregate structural differences, and we tend to think not. Okay, given the fact that the water contents at the time of sampling were not that different, the way we process the samples were not that different. And so I'm going to postulate that maybe there is less of a rhizosphere effect in the diversified cropping system. Okay. And so what we wanted to look at is, well, what constitutes this difference in terms of microbial biology? Okay. So when we look at these, this is a DSEC plot where we basically look at everything relative to bulk soil. So when you have a positive value, that means it's more abundant in the rhizosphere soil. If it's a negative value on this bar, that means it's more abundant in the bulk soil. Okay. And what we find is that there's very few different differentially abundant OTUs in these types. In fact, very few differentially abundant OTUs in general, particularly in the diversified system. But when you, my heavy hand again, but when you get to the vegetative stage 11 and the kernel development stage, you can see that you're up to 80 differential OTUs. And hence, that's why it's statistically significant there. And what we see is a big change in the proteobacteria, one of the more dynamic microbial groups in terms of metabolic activities. You also see a difference in Bacteroidetes and the chloroflexi and the Vericomicrobia. These organisms are known for their ability to degrade complex organic polysaccharides and molecules. And it's possible that they're being stimulated by the secretion of these polysaccharides by the root, okay? And they're increasing or differentially changing the abundance depending upon which group is most fit under that particular scenario. And we see some differences in other groups, actinobacteria and acidobacteria. Okay, and this is with using a fairly conservative Q-score. And if you do transcriptome data, you know you have to do, you can't rely just on your p-value, you have to adjust for a false discovery rate. Okay. So what we think is that, or what we suggest, is there's a real difference between these two in terms of how the cropping system influences the assembly in the rhizosphere. The question is, why do we not see a detectable rhizosphere effect with the bacterial communities in the diversified cropping system? Well, the thing that was even more apparent was how different the rhizoplant communities were. Okay, and do you remember from that those coordination plots, they're really off, separate from everything else, and they're very spread out. Okay, and when we look at that, we say, wow, at vegetative stage 4 relative to the bulk soil. There's 705 operational taxon units that differ between bulk soil and rhizoplane. Well, we can take some things here. There's some certain taxonomic groups of interest, but we simplified it. Rather than looking at the individual operational taxonomic unit, we asked, are there certain taxons that are particularly noticeable that are differentially abundant? And we looked at the family level, such as you'd be able to classify, for example, Nitrosimoniales or Rhizobiaceae into these different groups. Groups that we can associate a little bit with more physiological function with those taxonomies. And we did that, and I lost it. Okay. On the, behind here somewhere is, if I can, just need to send it back. Send it back. There we go. Sorry about that. That's so we broke it down to the family level, and what we can see is that the biggest difference, even at the family level separation, was at vegetative stage 11. Okay, a little less changes at other developmental stages, but notably, what I found particularly interesting was that if it's a reddish color, that means it was more abundant in the conventional cropping system. If it's blue, it's more abundant in the diversified cropping system. You find that there is certain taxonomic groups that change, particularly the Rhizobiaceae. They are much more abundant in the conventional system than they are in the diversified system. You also see a lot more abundance of certain groups of proteobacteria, the Bocarderiaceae and the Xylobacteriaceae, different beta proteobacteria that have been associated with plant growth promotion. Okay? And then there's also a lot more actinobacteria that are much more abundant in the conventional system. Actinobacteria would be things like Streptomyces. They're the organisms known for producing a lot of antimicrobial agents. Okay? And then, but if you look here in the diversified system, once again we're finding organisms such as Nitrosphere, which is a group of organisms, what we refer to as Kamamat, capable of oxidizing ammonia all the way to nitrate in the same organism. We also see a few actinobacteria, but a lot of Bacteroidetes. These are organisms that degrade complex organic material. Okay? And a few of the Vericomicrobia are much more abundant. As I mentioned before, the Vericomicrobia are an organism that has been identified as being lost through agriculture. Okay? That it seems to really decrease dramatically upon agricultural activities. So we also saw similar changes with the fungal communities. Once again, we saw that their biggest changes were actually at a vegetative stage 4 for the most part, and a little less so in the middle stages. If I was to show you the individual OTUs, you would see a similar trend, that there was actually a lot more individual OTUs here, and then they decreased in numbers of differences over time. It's a gender in their plant developmental stages, and that their dynamic properties diminished over plant development. So I'm going to summarize a little bit of Cassie's work. Is basically kind of saying the opposite. There's distinct prokaryotic and fungal communities defined by proximity to the root. Okay. The cropping system influenced bulk soil, prokaryotic, and fungal community structure, which really did not change appreciably over the growing season. And what I didn't highlight is that the ammonia oxidizing thawum archaea are much more abundant in the conventional system than they are in the diversified system. Okay. And that doesn't change over the growing season. And there are more Vericomicrobia and Bacteroidetes in the diversified system. And the thing that I'm still kind of grasping with is there a why did we only observe a rhizosphere effect, or detect one, at a period of high nitrogen demand? And is this a consequence of greater rhizodeposition by the plants in the conventional system? Okay. Don't know. Is it possible that the diversified system's prokaryotic cell microbiome is inherently more like a rhizosphere microbiome? One of the things that defines the diversified cropping systems is you've had an increased input of carbon through the of alfalfa, the oats, the soybean, as well as the composted manure. So maybe inherently it's more rhizosphere-like, so any exudation from the plant is less dynamic, less influential on the microbial community because they're already there, they're going to consume it quickly, and they're going to be competing with each other for it because they're already there. And I kind of look at this as kind of the reverse of a kind of a form of the camouflage hypothesis, which was used to understand biological control or biocontrol agents. A biocontrol agent can actually alter the rhizosphere to look more like a bulk soil rhizosphere, and then it masks the presence of the root from a pathogen. It's camouflage. Okay. And then I'm going to skip on to some other stuff. I'm going to talk also about the work done by a postdoc in my laboratory, Guillaume, where he talked doing the same basic questions, but it's all rhizotron based. Okay. And we allowed for much greater sampling numbers. We'd have n equals 16 at one plant developmental stage, and allowed us to go a little more deeply into looking at metabolic activity, active community members versus the total resident community. And one of the first things we noticed while we pulled the plant out of the rhizotrons is that the roots in the diversified system were much more ramified, much finer. So those soils were packed to the same bulk density. Okay, they were watered to the same level. And so it's possible that the root architecture inherently of a diversified system is going to differ, and that's going to promote in itself a very different rhizosphere and a rhizosphere effect. Okay. And so we have to always keep that in mind when we think about all the other results that we get. And when we do the ordination plots, this is just to highlight that when you look at the cDNA, this is all the cDNA here, or the RNA. This is our DNA. They separate. They're very different communities based on the ordination plots. And once again, in this case, we have bulk soil. We have rhizosphere soil right here, and then we have endosphere, what's inside the plant. In this case, the rhizosphere is really plants taken out, any loosely adhering soils retained, it was washed off with what's attached to the root. Okay. And so one of the questions we asked is, well, let's look at richness. Okay. And one of the things that was obvious to us is that the endosphere richness is much lower in the conventional system. Okay. 2,300 versus here. So that's one thing we know. So it's just a little less rich. That's basically species richness. When we look at the metabolic active, we see a real difference in the metabolic active members in these two different systems. What we find is that in the conventional system, it's always much lower in the conventional system, suggesting that there's not as many species stimulated to be metabolically active compared to the diversified system. So inherently, the diversified system might be just much more metabolically active, and you can see that there's really no statistical difference between these. Okay, and when you look at the diversified system, whether it be the bulk soil, the rhizosphere soil, or even the endophytic community. Okay. And so I have to give my obligatory phylum level bar chart, which really is hard to interpret. Okay. That is, basically shows, if you're looking at them, well, you look at the black bar in particular, those are the pseudomonads, one of my favorite organisms, okay? And you can see there's a real dynamic change of relatively 20% in the soil, upwards to 40% in the rhizosphere, and they increase up to 50-60% in the rhizosphere. Whereas the yellow one is the Thalamarchaea, ammonia oxidizing organism, fairly abundant in the soils, but they're actually statistically lower in the diversified system, both in the soil as well as the rhizosphere as well. And you can see that some different groups are increased. The microbia are much more abundant in both the diversified soil and rhizosphere. Okay. And some of those trends go all the way to the rhizosphere. So let's look at this a little bit more deeply. And this is a tough diagram to look at, but what we're looking at here is we're just looking at ribosomal RNA gene abundance. We're looking at the total diversity, okay? And we're comparing the rhizosphere of the conventional system, which will be in red. So if the bar goes out, okay, in red, that means it's much more abundant in the conventional system. If it's green, it's much more abundant in the diversified system. Okay, and what you can take home is that there's differences there. If there was no difference, there would be no outward radiation. Okay, and sometimes you see no outward radiation because there's no statistically significant difference between different taxons. Okay, and you can look at the relative abundance of these groups as well. You can see, well, they look fairly similar on the DNA level, but when you get to the RNA level, you can start seeing that there's some shifts. Okay. And for in this case, for these taxons here. So I want to then go to a different way of looking at our information. Okay, how do we know who's metabolically active versus who's not? Okay, you could have 10 organisms with no ribosomes, and one organism with 10 ribosomes, and you'd have basically different pieces of information here. And so what we did is we basically did the same type of analysis. We compared pairwise contrast between our metabolic active, this is the 16S RNA, our green, the red is DNA. So if it's red, it's more abundant, there's more DNA there relative to RNA. Okay? And then what you see is that there's some distinct differences. This is the conventional cropping system. This is the diversified cropping system. And you can see there's a lot of similarity. Okay, so we've actually narrowed down. We're saying that they're a little more similar than we would have thought earlier. Okay, but there are some hallmark differences, particularly in the proteobacteria. There's a group of alpha proteobacteria that are now much more metabolically active in the rhizosphere in the diversified system that's not present here in the conventional system. You also have you have a broader array of actinobacteria more metabolically active as well. Okay? And so what we can do is look at this bar chart, just at the alpha proteobacteria, to get a sense of, okay, all I've said is that they're different. It doesn't say what the relative abundances are. Okay? And this bar chart just shows us that one thing that's really different in the conventional system is the presence of rhizobiaceae. Okay? But they're not very metabolically active relative to the amount of RNA we get there. So there's a lot of residence there, but at the time we sampled, they weren't really metabolically active. What we did see is certain groups of organisms, like the Caulibacter and also the Rhodospirales, were all much more metabolically active relative to the amount of DNA there. Well, nothing more than a group of nodes, well connected to each other, but less so with others. Okay. And then last is Keystone taxa. That is OTUs that, if removed, collapse either a module, or, if it's truly the linchpin of Keystone, the entire network. Okay. So we did that. We did that in soil, the rhizosphere. And what we found is, in general, the microbial networks in the diversified system are much more interconnected. Okay? And if you look at the rhizosphere, you can see that it was comprised in the conventional system of about 133 nodes versus 269 in the diversified system. The number of links, 847, that means positive and negative linkage associations between the different members. 847 versus 1941. Okay? And the same is true for the endosphere. In fact, when we look at the endospheres of these two same plant grown in these two different soils, what's inside the plant is different, partly because of who's outside to get into the plant is different. Okay, but there is some narrowing selection as it enters the plant. So we found also, when you look at the metabolic active community, it's even more interconnected, obviously, than it would be if you look at just DNA. Okay, and it's generally true both for the soil, the rhizosphere, and somewhat for the endosphere. And what we can do is basically parse out who's there. Okay, can we glean any information about who potentially is interacting with each other to develop experiments to test whether or not these organisms are important for a particular co-interaction. Okay, and a couple things that come out is that many of these organisms involved in these modules, okay, and we're just doing something simple, the endosphere, okay, anything that's underlined means it's not in the proteobacteria. So the proteobacteria, obviously when you look at the bar charts, 60% of the endosphere was dominated by proteobacteria. And obviously they play a major role in their interactions. But there's some unique interactions, particularly some that are just modules of just proteobacteria and Comomonas. Those are actually phosphate solubilizing organisms, typically. And others are a mixture of different taxa. And then we wanted to look at a little bit more about the network properties of the community. And we can actually look at connectivity within a module and connectivity between modules. We get a value, we plot it, okay? And if using some previously published values, we can then select an area that we'd say was, well, if they fall in this area, if they are among module connectivity, they're positive, or they're connectors. If there's a lot of within module, they're module hubs, okay? And if they have both attributes, they're a network hub. And what we found is that this is really what we saw before, and when you looked at it pictorially, we have now some mathematical validation that the connectivity is usually larger in the diversified system. The path length is shorter. Okay. Same is generally true for the rhizosphere. Not so much with the endophytic communities. And then we can identify the number of connectors and module hubs, and we actually were surprised to find actually one network hub. Okay. So then the question is, well, was relatively surprising because not too many studies have been able to identify module hubs or network connector hubs in microbiome nets, and partly because of sample size. Okay, we had 16 for each condition because we knew you need that kind of power to be able to ascertain whether or not you have potential interactions of that nature. And so what we did, these are a series of pie charts, okay, that by color associate with the connectors and hubs, okay, the relative percentage of them. So in the module hub for the soil, conventional, it's got 18 hubs, and there's a proportion that are Thaumarchaea. And there's another portion bar that sticks out the dark blue, that's Nitrosomonaceae. And I'm going to argue there is overrepresentation of ammonia oxidizing organism as keystone taxa. They comprise anywhere from the lowest around 15% or 10 to 15% to greater than 30% of the module hubs or connectors. And if you look at that, if you look at all the groups of organisms there, these organisms, surprisingly, I would not have anticipated being potential hubs for taxa. But then on the other hand, ammonia oxidation is the keystone step in the nitrogen cycle, okay, and nitrification in particular. It basically controls the flux from ammonia to nitrate. And you might see differences between these different systems based on the nature of the different cropping systems. And to ground truth it, get to something that's more practical, we also looked at our potential metabolic activity in these systems. And the take home is that there's higher potential nitrification. This is where you give an excess substrate and see what happens in the bulk soils. And they're not really in both the bulk soil and the rhizosphere soil. So we actually isolated rhizosphere soil and did an activity assay. And they were basically the same level. However, the diversified bulk soil was lower than the conventional bulk soil, and even lower yet was the potential activity in the rhizosphere of the diversified system, which might then contribute to the reductions in nitrate leaching from the diversified systems. And you know what, given the time, I'm going to skip over these and just basically say the ammonia, our quantitative PCR validated what we saw in terms of the abundance of ammonia oxidizers, both archaea and bacteria, and it correlated actually with the nitrification potential activities. And when we look at whether or not there are particular operational taxonomic units that were overly abundant, that were keystone species, they're not. So sometimes we have a keystone species, this could be a module hub, it's a very small proportion. So they don't have to be numerically dominant to be involved in a network or a keystone or a module hub or connector. And I think it's pretty self-explanatory. We've demonstrated there's differences between the cropping systems. There's differences between the different levels of proximity to the root. And there's different ways of looking at the community based on whether you look at it as metabolically active or total residence. And also, I think the area that we'll explore even further is more in the microbial networks to allow that. Can we use this to understand rules of assembly, or as well as physiological functions that are important for plant health and nitrogen cycling? And we have a lot of ongoing work, but I won't talk about it because we're running out of time already. So, any questions? I know I went through that fast. Yeah.
My question is kind of a vague one, but we're also kind of analyzing networks, and when you think about plants and animals, if species are positively or negatively correlated, a lot of times it's habitat preference and not competition. Like if it's negatively associated, they just prefer a different habitat. Mm-hmm. And I was just wondering, when you think about bacterial networks or fungal networks, is that part of it or not? Or do you think they're associated with a metabolic pathway or something?
Both. I mean, there are clearly bacteria that prefer certain microhabits, and that's a physical environment.
Yeah.
And some of them leads into a metabolic codependence. So there could be a syntrophic interactions. So it's a little bit of both, but our scale's pretty crude. I mean, the way we pull out the soil is really not really reflective of the microbial habitat scale, whereas you've got more realistic scales or a more truth scale. So it's hard to say what's the habitat scale there. So I don't have a good answer for that. Yeah.
You call them Brady diagrams or something like that, where you had the bacteria and the fungi, and the bacteria looked like they were more diverse than the fungi. Can you go back to that?
Yeah.
Kind of early on.
Yeah, where we have the coordination plots of dissimilarity between the bacteria from our samples. And it'll be, there we go.
Yeah. Okay, so I think I heard you say there's the prokaryotes on the left and fungi on the right.
Right.
The fungi weren't diversifying to the extent that the bacteria were?
It's a little more complicated than that. The fungal, the bacterial, when you look at rhizoplane, diversity comparing to rhizoplanes, is they became they became more alike on the rhizoplane when the plant aged. They became more similar. So, because the rhizoplane here, it's 0.056 difference. This was
4 developmental stages. You just lost it.
Oh.
So there's 4 developmental stages there, and you can see some significant trends among the bacteria, but not the fungi, right?
Well, you see it in both the bacteria and the fungi both. Okay. But you see them a little differently. There seems to be that when we did the comparisons between here, these were more dissimilar than these were when we did contrast the rhizosphere. Both, particularly in the soil and in the rhizosphere. So particularly the rhizosphere. So if you look at the rhizosphere here, you see how, in the bulk soil, see how far apart these are relative to these right here. My heavy thumb. Well, I went a long ways, didn't I? Okay, so when you look at here, you see the red. Okay, the red to here, the 2 different symbols. It's hard to see, but this would be the conventional. This would be the diversified. Oh, excuse me, conventional, diversified. They're fairly separated. When you look over here, you can see that they're almost right on top of each other. They're still statistically different, but they're much greater apart here.
I missed the point then, because I couldn't discriminate between the triangles and
Yeah.
the circles. Okay. Thank you.
Mm-hmm.
Following up on that point, do you see a difference in fungal functional types like white rot, brown rot among these different systems?
I'm going to have to get a better fungal biologist than myself and Cassie and others. We do see some. Really, the big changes are the ascomycetes. The biggest change. There's some bacillomycete changes, some zygomycetes, but primarily it's ascomycetes. Yeah.
So if I understood you correctly, the ammonia oxidizers were less abundant and doing less work in the more diverse systems. Is that correct?
Correct.
Okay, so the more diverse systems had corn plants that were larger and took up more nitrogen.
Mm-hmm.
How did that happen?
It's either the bacteria are better at turning over organic nitrogen that the plant can get to, or there's nitrogen fixation occurring, or a combination thereof. I can't say either what it is for either one of them. In the ideal world, we would have seen a shift towards known free-living non-symbiotic nitrogen fixers in the diversified system. And eureka moment, and it would be easy to explain. It's not that simple. Because nitrogen fixation is really prevalent in a lot of different groups of organisms. And it's not just the rhizobia. No, methanogens do it. I mean, all kinds of organisms do it. And so it could be coming, if that's the contribution, it could be from a way that we're not aware of. Was that obvious to us? There was another hand. Yep.
So, do you think this is solely due to developmental stage of the corn plant, or is it also compounded by different soil moisture, soil temperature? So if you staggered your planting, so you're sampling at the same date for all the stages.
Well, they all are basically this. They're not that different in the stages when we sample.
No, so that you sample on
Oh,
one
stage.
day,
Oh,
but
you're
they're,
saying-
you know,
oh, yeah.
before on the same day as the fruit plant.
Okay, what I didn't show is if you look at the microbial community of the soil over the growing season, they don't change statistically.
Bulk soil?
Bulk soil. You actually see a little bit more in the fungal community changing over time. It changes initially very quickly, then it stops. That might be because it could be that the changes are not, there are changes. There's a small number of OTU changes, but they're not dramatic enough based on the census to say, based on beta diversity, that they're different. Okay. There are some taxonomic groups that differ over time, but they're not enough to say that the community on a beta diversity level is different. That's the problem with community descriptions. What is the right descriptor? But that's a very good question. And we were surprised. We thought we would see more seasonal changes in the bacterial community. And there are some shifts. When you're to plot them right next to you, you do see some shifts. But they're too small, really, to be significant statistically anyway.
And that's true for RNA too, or for DNA?
That is true for DNA and RNA. Well, actually, we didn't do RNA, excuse me. We did not do RNA in the field. We only did DNA. RNA was very hard. We had to have so much material. and we had to be very conscious of losing, of being taking too long and losing RNA in our sampling process.
So it's possible that it didn't change because, I mean, the same microbes are there, but they're active at different times.
Right. I think really what I'm after this experience, I would argue we should be really doing out in the field is really looking at the metabolically active community over time in the growing season, and I bet you we would see differences. We don't see it in the resident population. That's not dynamic enough. And there's a lot of, the problem with the DNA is there's a lot of relic DNA, dead cells, spores, and you amplify those. You know, they turn over, but they don't turn over that quickly, okay? Whereas RNA at least gives, you know, the problem with RNA is, no, a spore has maybe 1 or 2 copies of the genome, and it actually has quite a few copies of the ribosome. It's still not metabolically active. Okay, so you have to take everything with a bit of perspective. Any other questions?
Thank you.
All right.